Hi, my name is Lee Lind, and it's my pleasure to welcome you to the Jones Seminar Series at the Thayer School, obviously. Today's speaker is Professor Dan Olson. It's my pleasure to introduce. Dan was a Dartmouth undergraduate, majored in physics, went to work for a period of time for a startup in the area, Mascoma Corporation, in the biofuels field, came back to Thayer School for a PhD, and for the last 10 years or so post-PhD has been leading research, not only here, he leads metabolic engineering research, which you'll hear about, here as a member of the Department of Energy-supported Center for Bioenergy Innovation, which used to be called the Bioenergy Science Center, a very large consortium that Dan will describe. But he also leads a metabolic engineering group at the University of Campinas in Brazil. He has been an author on over 70 papers, and his work is supported by the Department of Energy and the National Science Foundation. So please join me in welcoming Dan, and I'll get out of the way.
Thank you. Today I'm going to be talking about engineering microbes for cellulosic biofuel production. And so first, I'll explain a little bit about these pictures. So these first three pictures here, this is cellulose. Cellulose is the fibrous, woody material that makes up plants. This down here, this is a picture of my favorite bacterium, Clostridium thermocellum. It's the bacterium is these yellow cells right here, and they're growing on the green false-colored cellulose, but that's all false-colored electron microscopy. And here you can see a metabolic pathway. This is a schematic diagram of the reactions inside of the cell to convert cellobiose to ethanol. So I'd like to start out by acknowledging my colleagues and collaborators. All research is a team effort, and I really want to take a moment to recognize these people. I want to particularly thank the postdocs, technicians, and the students I've worked with. These are the people who actually performed the research. Without their years of hard work and dedication, this would not have been possible. Getting ideas into the real world requires diverse expertise, including simulation and evaluation, commercialization, upstream processing, and also downstream processing. The bioenergy group at Thayer is unique in that it brings this expertise together in a single place. There's no other group in the world that does this. My area of expertise is metabolic engineering. Taking organisms that naturally break down lignocellulose and converting them into efficient biocatalysts that can withstand the rigors of industrial scale fermentation. Outside of Dartmouth, I also lead a metabolic engineering group in the Advanced Second Generation Biofuels Laboratory at the University of Campinas in Brazil. I lead the Pathway Engineering Group at the Center for Bioenergy Innovation, funded by the U.S. Department of Energy. I consult with the TerraGeo Corporation about strain development, and I collaborate with other researchers around the world. Today, I'm going to cover three topics. First, I'm going to explain a little bit about the need for cellulosic biofuels. For many technologies, the people studying the need for the technology and the people getting the technology to actually work only occasionally talk to each other. However, a unique feature of the bioenergy group at Thayer is the presence of both of these activities in the same place. Second, I'm going to talk about several recent improvements in cellulosic biofuel technology based on my own research. And finally, I'm going to explore the broader implications of my work on the field of metabolic engineering. In this part of the talk, I'll explain why the world needs cellulosic biofuels. Stabilizing the climate requires that we eventually reduce net human-generated greenhouse gases to zero. This graph here shows the rate of CO2 production as a function of time. The area under the graph represents the accumulation of CO2 in the atmosphere. So to get from business as usual to the below 2 degrees C warming scenario, we need to dramatically reduce CO2 emissions. That's indicated by this green square here. Reducing emissions involves things like replacing fossil fuels with biofuels, switching from fossil fuel consuming devices to renewable electricity, and improving efficiency of various energy consuming devices. However, even with aggressive measures to reduce emissions, there are some industries that are hard to decarbonize. As we pick the low-hanging fruit, it gets progressively more difficult to make further progress. This is why we also need CO2 removal technologies. Sometimes called negative emissions, these are technologies that remove CO2 from the atmosphere and potentially allow it to be stored underground. These technologies can offset the emissions from the hard-to-decarbonize sectors, such as transportation, and allow us to achieve net zero CO2 much faster. To achieve net zero emissions, we need both reduction and removal technologies. Getting to net zero CO2 requires contributions from a variety of sectors and technologies. This graph here shows the rate of CO2 emissions in 2005. You can think of this as a vertical slice of the previous graph I just showed. Note that the numbers are about tenfold lower because this is data only from the United States, whereas the previous graph was global data. The stacked subbars, so these stacked subbars show several scenarios for reducing CO2 to zero, broken out by industry. And the shaded areas at the top of the different bars represent our relative uncertainty about their contribution. Biofuels can contribute to decarbonizing the transportation sector by replacing fossil fuel carbon with carbon that was originally captured from the atmosphere by photosynthesis. Although there are a variety of ways to do this, you can see that all of these scenarios include a substantial contribution for biofuels. The heavy-duty transportation sector is one area where the need for biofuels is greatest, due to the requirements for high energy density. This graph shows energy density both by volume and mass for a variety of different energy carriers. Liquid hydrocarbon fuels have orders of magnitude higher energy density compared to batteries. This graph shows the mass energy density of batteries over time. So you can see that over time, battery technology has been improving. And it compares the energy density over time versus the requirements for aviation. And you can see the requirements are different based on the scale of the plane and the distance the plane needs to travel. Although electrification of small short-range planes may be possible relatively soon, electrification of the large planes and the long-range flights that account for the bulk of the emissions from this industry are still several decades away. This graph shows a plot of energy usage from the transportation sector broken out by energy carrier. The 2010 data is a base case, and the future predictions are based on a 2-degree scenario. Now, just to explain this jargon a little bit, in the field of climate modeling, there are a lot of different factors that can go into, you can choose to reduce one technology or increase another technology, and these get lumped together in these things called scenarios. So early on in the talk, I mentioned a 2-degree scenario. What a 2-degree scenario means is a set of assumptions that will give a reasonably good chance of maintaining global warming below 2 degrees. And a shorthand way of saying that is a 2-degree scenario. So biofuels are one of the few low-carbon options for the heavy-duty transportation sector, primarily due to this reason of energy density. Ethanol is an important molecule for liquid biofuels because it sits at the intersection of several important properties. It can be produced biologically at high yield and concentration, and its low boiling point makes it easy to separate by distillation. Furthermore, ethanol can be chemically upgraded to longer-chain fuels at low cost. Several companies are currently working on this. It's referred to as ethanol to jet conversion. And in the past 2 years, there have been over 50 purchase agreements for this type of fuel. In addition to displacing fossil fuels, biofuels enable cheap long-term storage of CO2 that's been collected from the atmosphere by photosynthesis. So that is this part of the graph, the negative emissions part. Although this potential was overlooked for many years, it has recently attracted more attention. This graph shows projected primary energy supply by category based on a median of 85 different global warming scenarios, 1.5 degrees C global warming scenarios, through the year 2050. You can see biomass is predicted to play a larger role than solar and wind combined. Now this graph over here shows a comparison of several carbon removal technologies. The primary axes are energy requirements and the total capacity for negative emissions. Along the top of the graph here, we can see the three negative emission technologies with the highest capacity for negative emissions. They are direct air capture, or DAC, afforestation, or AR, and bioenergy with carbon capture and storage, or BECCS. Bioenergy is frequently included in climate change scenarios due to its unique combination of low energy requirements, low cost, and high capacity for carbon removal. How do biofuels allow for carbon capture? During photosynthesis, green plants capture CO2 from the atmosphere and fix it in green plants. These are then collected and brought to a cellulosic ethanol facility. About 30% of the carbon that's collected is converted to ethanol, where it can be used to displace fossil fuels in vehicle transportation. When that fuel is burned, the photosynthesis-derived carbon is returned to the atmosphere as CO2. And you can see that here, sort of, completing this CO2 cycle. About a quarter of the carbon is released as a high-purity CO2 stream from the fermentation off-gas and anaerobic digestion of waste products. This CO2 is currently released to the atmosphere, but its high purity makes it cheap and easy to collect and store underground with minimal processing. And in fact, if we wanted to maximize carbon storage, it's possible to capture additional CO2 streams. In total, up to about 70% of the photosynthesis-derived carbon can be captured, highlighting the opportunity of cellulosic biofuels to make a substantial contribution to negative emissions. Here, I will describe the carbon flows in biomass in a bit more molecular detail. During photosynthesis, the Calvin cycle is used to capture carbon dioxide from the atmosphere. 6 CO2 molecules are captured to create each sugar molecule. Next, each sugar molecule is broken in half in glycolysis to create pyruvate. Finally, the pyruvate in glycolysis is fermented to ethanol, resulting in the release of 1/3 of the carbon atoms as CO2. The release of CO2 is a fundamental part of the fermentation process, something that you might be familiar with if you've ever drunken beer or champagne, which is where the carbonation for those beverages comes from. The CO2 released during fermentation has often been thought of a waste product, since it doesn't end up in the resulting fuel. However, in the context of carbon capture and storage, the CO2 stream turns out to be highly beneficial due to its high purity and ease of collection. Another way of saying this is previously we've only thought about biofuels for their ability to provide the actual liquid fuel that we want for transportation. But in fact, the fact that biofuels are collecting carbon for us turns out to be a significant additional benefit, which before we totally ignored and now is a substantial driver of this process. There's a clear need for biofuels, both to decarbonize the heavy-duty transportation sector and for low-cost CO2 removal. How do we make this happen? In the next part of the talk, I'll describe some recent advances in cellulosic biofuel technology. Lignocellulose is a complex material made of cellulose, hemicellulose, and lignin. This diagram shows the structure of biomass at progressively smaller scales. So here you can see the plant, and then inside the plant are plant cells. Inside the plant cells are fibers of cellulose, which have a combination of lignin, hemicellulose, and cellulose. At the core of each of these fibers is a cellulose, crystalline cellulose fiber. That fiber can be broken into cellulose chains. Each cellulose chain is a polymer of glucose molecules, and those polymer chains of cellulose can then be broken down into individual glucose molecules. The fermentation of lignocellulose can be divided into 2 parts. The first step is solubilization. So solubilization is this first part, going from something that looks like a plant growing in the ground, breaking it up and converting it down into something soluble that can be taken up by the cell. The next step is product formation. This involves the conversion of these fermentable carbohydrates into useful products such as biofuels and ethanol, for example. The current paradigm for lignocellulose deconstruction involves yeast supplemented with a cocktail of several fungal cellulase enzymes. In this diagram, you can see this big yellow thing over here. This is the yeast cell. These green chains, these are cellulose chains, with each unit being an individual glucose unit. And here you can see individual enzymes cleaving off chunks of glucose polymers. And here's a photo of what that looks like in the real world. You can see that the yellow is the yeast cells. This red thing right here is a cellulose cotton thread, and the enzymes don't actually show up on this image. Organisms that specialize in cellulose fermentation have evolved over billions of years, and they do things much differently. They use a cellulosome, which is a multi-enzyme complex with hundreds of different cellulase enzymes connected in a hierarchical structure. The number of cellulase enzymes is important because, as you saw from the previous slide, lignocellulose is a complex material with a diverse array of chemical bonds in various 3D conformations. In addition, cellulosomes perform two key roles. They physically attach the cellulase enzyme complex to the cellulose surface, and they also attach that to the cell. And this is a photo of what that looks like. So here you can see the same cellulose thread, and now you can see these enzymes, or sorry, not the enzyme, the Clostridium thermocellum cells. And you can see that almost every available piece of surface area is covered by these cells. As a result of these natural advantages, native cellulose fermenting microbes are much more efficient at fermenting cellulose. Since cellulose solubilization is the rate-limiting step in cellulosic biofuel production, we believe that starting with nature's best organisms is critical to getting this technology working. The classic approach to cellulosic biofuel production traces its origin to beer and wine fermentation and uses yeast as the fermenting organism. Since yeast can natively solubilize cellulose, this requires thermochemical pretreatment and added enzymes, both of which add to the cost of the process. Native cellulose fermenters are very good at consuming cellulose, but do not natively produce ethanol at high yield or concentration. Based on our current state of knowledge, robust cellulolytic ability seems to be a more difficult phenotype to move compared to robust ethanol production. And this is why my group has focused on engineering native cellulose fermenting organisms. My research to date has focused on three areas: improved genetic tools, metabolic pathway development, and microbial physiology. In the next few slides, I'm going to give you a few examples of some of these types of research. DNA is the primary means of information storage within a cell. Therefore, if we want to reprogram cells, we need to change their DNA. Electroporation is a technique for inserting DNA into cells. Application of an electric pulse creates a field which creates small holes in the cell membrane. If the conditions are right, foreign DNA can go inside the cell, and then after the electrical pulse is over, the holes close up, allowing the DNA to be transferred into the cell. This technique requires a combination of knowledge from the fields of both electrical engineering and biology. When I first started working on this project many years ago, I worked with Professor Charlie Sullivan to build a high voltage electrical pulse generator. So you can see here's the relatively simple schematic of what this device looks like. It's really just a capacitor and a switch and a device to turn it on and off. And here's a picture of what the thing actually looks like. It turns out that in practice, building a high voltage pulse generator is non-trivial, both due to the real world effects. You get leakage currents in various circuits that are not designed for in the schematic, and also just the practical applications. Working with thousands of volts of electricity is not super safe. There are a lot of safety concerns to deal with that safely. And thankfully, working with Charlie Sullivan, he sort of walked me through all that and helped me avoid some of the deeper cliffs to fall off of. It took 3 years to get C-thermocellum transformation working reliably. The critical step in the process turned out to be the recovery temperature. By accident, when I was doing these transformations, I discovered that putting the tubes where I was growing my cells on a different shelf in the incubator gave different results. It worked on one shelf, it didn't work on the other shelf. Now in an incubator, you would expect, the incubator, the whole point of an incubator is the temperature is the same all the way through the incubator. However, if you put a thermometer in your incubator, you'll actually find that there are differences. The different shelves are a little bit different depending on where the fan is blowing or the arrangement of the items inside. Well, it turned out that one shelf is a little bit warmer than the other. And when you do this experiment on one shelf, it works, and when you do it on the other shelf, it doesn't. And so when I investigated this in more detail, I realized what was going on. You can see that the transformation efficiency, as the temperature increases, the transformation efficiency, it goes up and up and up, and then it falls off a cliff. To this date, I still don't know why that works. However, every group in the world that I've trained to use this technique, that's kind of the key point. I say, look, measure the temperature, put 3 thermometers in your incubator so that you know the temperature is right. And then when they tell me it doesn't work, I tell them to put 1 more thermometer in, and that usually solves the problem. But anyways, to date, this technology is now working in dozens of labs around the world. And basically everybody who is working with this organism is now doing it using this technique. Once we had figured out how to get plasmid DNA into the cell, we needed to figure out how to modify the chromosome. Homologous recombination is a naturally occurring process that organisms use to repair breaks in DNA. During the repair process, part of one strand of DNA is copied onto the other strand. We can co-opt this process to make targeted modifications on the chromosome. By constructing synthetic pieces of DNA with regions that are homologous to the chromosome, we can trick the organism into inserting or deleting specific DNA sequences. So in this diagram here, you can see we construct a plasmid DNA. A plasmid is a piece of DNA where we specify each individual letter of DNA along the entire plasmid. And these blue regions represent the homology regions. And then through a series of recombination, we can encourage the organism to integrate that plasmid, or rather parts of that plasmid, into the chromosome in exactly the configuration we want. Now, one of the interesting things about homologous recombination is each homologous recombination event is, a 1 in 1,000 event. 2 homologous recombinations together, which is what you need every time you make a genetic modification, is a 1 in a million event. However, when you're working with billions of bacteria, we get 1 in a million events all the time. And now this technique is working, and reliable and iterative chromosomal modification of C. thermocellum is routinely done here at Dartmouth. After developing tools to engineer C-therm, we needed to decide what modifications to make. In metabolic engineering, one of the key metrics is yield, the amount of product produced per substrate consumed. For example, one Cellulose molecule can be converted into 4 molecules of ethanol. A standard approach in metabolic engineering is to delete pathways for competing products. If the organism wants to grow, it has to produce the desired product. We pursued this strategy for C-therm and deleting pathways for several competing products, including acetate, lactate, formate, and hydrogen. This graph shows the result. The strain was successfully engineered to convert Cellulose into ethanol at about 75% of the theoretical maximum yield. For commercialization, C-therm needs to meet certain process metrics for ethanol yield and concentration. The commercialization space here is shown as a green box. The lower boundary of the box is determined by operating costs, which increase at low product yield. The left and right boundaries of the box are determined by constraints related to either separation costs or solid loading. The improvements in C-therm ethanol production so far place it at the outer edge of the commercialization space. Further improvements in product concentration are likely needed for commercial application. Having succeeded at engineering C-therm for increased ethanol yield, the next challenge was to increase product concentration. One factor that can limit product concentration is tolerance. Growth-linked selective pressure can only operate up to the maximum product concentration an organism can tolerate. Using growth-based selection to further increase product concentration requires an increase in tolerance. Using ethanol addition experiments, we observed the maximum ethanol concentration of engineered strains appeared to be limited by tolerance. Previously, we had shown that in wild-type seatherm, we could adapt that organism to tolerate ethanol up to 50 grams per liter. So we decided, okay, if this works in the wild-type strain, let's see if we can get it to work in the engineered strain. When we adapted engineered strains of seatherm for faster growth in the presence of added ethanol, we were hoping that at least some of the mutations would increase both product tolerance and final concentration. Instead, we found that ethanol production had decreased, and this decrease was one of the most consistent observations that I've observed in all of my strain adaptation experience. Basically, if you grow a strain of Seathermicellum in the presence of ethanol, you will find decreased ethanol production. When we resequenced the genomes of the adapted strains, we found a likely explanation for this effect. In all 11 of the adapted strains, we found mutations in a gene called ADHE. Furthermore, reviewing the literature data, we found mutations in the same gene from 2 prior studies of ethanol tolerance in seatherm. Measuring enzyme activity, we showed that these mutations had the effect of eliminating ethanol alcohol dehydrogenase, a key step in the ethanol production pathway. This explained why ethanol production had decreased in these strains, but the observed trade-off between ethanol tolerance and production suggested the need to develop a better understanding of factors that limit product concentration. In metabolic engineering, we usually start by thinking about stoichiometry, the ratio of substrates and products in a chemical conversion. This can help us understand yield, but not product concentration. In fact, the field of metabolic engineering lacks a systemic approach for improving product concentration. Thermodynamics, on the other hand, can give insights into product concentration. And by applying thermodynamic principles to metabolic pathway design, it may allow for a more systematic approach to this type of problem. One potential factor limiting product concentration is local thermodynamic equilibrium. In enzymology, it's common to demonstrate the function of an enzyme using a thermodynamic free energy diagram. That's what this diagram is right here. So here you can see the reaction coordinate going this way and the free energy on the vertical axis. Going from the uncatalyzed reaction is represented by the orange line. The enzyme catalyzed reaction is represented by the purple line. By adding an enzyme, you don't change the overall thermodynamics. The start and the end point are still the same. However, the reaction can proceed more quickly because the activation energy has been reduced. Recently, a similar framework has been developed to understand thermodynamic effects of metabolic pathways, known as the min-max thermodynamic driving force analysis, or MDF. We can plot the free energy change of a pathway using the same coordinates. So here we have, instead of the progress of the reaction, we have the progress of the entire metabolic pathway, which is a series of chemical reactions. And here we have the same free energy coordinate. Initially, we start with the reactions at standard conditions. So standard conditions means 1 molar concentration of products and substrates. That gives us the delta G0 value, which gives us this, this dashed line here, this gray dashed line. However, reactions with a positive delta G value will not proceed forward in the forward direction. Therefore, we need to adjust those values. So we can use, this is an example of an equation for just a standard chemical reaction with 2 substrates and 2 products. In this type of analysis, we have the delta G0 value is constant and the RT term is also constant. And therefore, we adjust the slope of these lines by changing the concentrations of the products and reactants. If there were no constraints on metabolite concentrations, we could evenly distribute the free energy across the entire reaction. And that would give us a conversion shown by this straight green line. However, in real biological systems, there are constraints on metabolites. There are constraints on the low end due to kinetics, and on the high end due to molecular crowding. Furthermore, there can be internal constraints if a single cofactor is used in multiple reactions. This is common for reactions that involve cofactors, for example. Taking these constraints into consideration gives us this purple line. A fundamental insight from this framework is that local thermodynamic bottlenecks can limit product concentration, even if the overall transformation is thermodynamically favorable. And what that would look like is, so here you have an overall transformation which is thermodynamically favorable, which you can see by the fact that this, you start off at a higher point, a higher delta G than you end up with at the end. What a local thermodynamic equilibrium or a local thermodynamic bottleneck would look like is if you try to take this dashed gray line and during your optimization you got stuck, where one of your segments was either slightly pointed upwards or horizontal. That's an example of what a thermodynamic bottleneck would look like in this MDF framework analysis. So here's an example of how this could be used to engineer product concentration. In wild-type C-thermocellum, two key reactions use the same cofactor, NADH. There's the gap-pH reaction in glycolysis and the alcohol dehydrogenase reaction in the fermentation pathway. One of these reactions is more thermodynamically favorable when the NADH levels are high. The other one is more thermodynamically favorable when the NADH levels are low. Initially, when fermentation begins, they can agree on an NADH level that works for both of them. However, as the ethanol concentration increases, it eventually becomes difficult for these two enzyme reactions to find an NADH level that allows forward flux for both of them. That is where you get a local thermodynamic bottleneck. However, if we want to fix this problem, we can change the cofactor specificity of the ADH reaction from NADH to NADPH. This breaks the thermodynamic linkage between the ADH reaction and the gap-pH reaction, potentially eliminating this constraint. Now, in thermodynamics, there's no such thing as a free lunch. So the additional thermodynamic driving force for the ADH reaction comes at the expense of something. And that something is the P4 reaction. So you can see right down here, there's this P4 reaction. And in our initial scheme, this has a thermodynamic driving force of negative 10.6 kilojoules per mole. So we steal a little bit of the thermodynamic driving force from that in this next scheme. But you can see that this is the overall thermodynamic driving force is limited by this part of the reaction. So even though the thermodynamic driving force of the P4 reaction is a little bit lower, it's still not low enough that it has become a bottleneck in the system. We are currently performing the protein engineering necessary to actually test this hypothesis experimentally, because in order for this scheme to work, we need to take the native enzyme, which is NADH linked for both of these reactions, and engineer the protein such that it can use NADPH for both of these reactions. And this gives an indication of the progress of that work. So here, for the ADH domain, we start off with an enzyme that's completely NADH linked. And over the course of a series of mutations, we have an enzyme, a mutant enzyme, that's over 90% NADPH linked. And then for the ALDH reaction, again, we start with an enzyme that's 100% NADH linked. And we were successful, although a little bit less successful. And now we have an enzyme that's about 50-50 NADH and NADPH. Now I will spend a little bit of time talking about the future directions of my work. Petroleum is currently one of the main feedstocks for the chemical industry. To decarbonize the chemical industry, we eventually need to replace petroleum refineries with biorefineries. For microbial conversion processes, product concentration affects both capital costs and operating costs. Thus, finding ways to engineer organisms for high concentration production is necessary to enable this transition. In the field of metabolic engineering, increasing product concentration is more of an art than a science. In many cases, there's a gap between the product concentrations reported in the literature and the concentrations needed for commercial application. Although there are many individual success stories, there's a lack of a unifying underlying principle to guide further work. One of my long-term goals is to develop the theoretical basis for limitations to product concentration. The diagram here is a flowchart representing possible chains of causation. My recent work is focused on the local thermodynamic equilibrium mechanism. Going forward, I'm interested in investing in investigating the other possible mechanisms, as well as mapping existing success stories from the literature onto these various pathways of causation. Eventually, this will allow for rapid diagnosis and remediation of product concentration limitations in a variety of organisms and products. One of the major problems standing in the way of understanding limitations to product concentration is the complexity of microbial metabolism. Much existing work has focused either on the very complex systems of whole cells or the very simple systems of individual purified enzymes. One of the ways I plan to untangle this complexity is by studying systems with intermediate complexity. This includes in vitro pathways, cell-free systems, and wash cell systems. As one brief indication of how this is going, my postdoc, Bilal Jilani, who is sitting right up there, recently constructed an in vitro pathway to convert acetone into 2,3-butanediol and achieved a concentration of 189 grams per liter, the highest reported to date anywhere. In conclusion, cellulosic biofuels provides two distinct ways to reduce CO2 levels, both by displacing fossil fuel emissions and also by allowing negative emissions. Over the past 2 decades, I have developed genetic tools for seatherm, used those tools to engineer this organism for increased ethanol production, and shared those techniques and strains with collaborators around the world to advance the state of cellulosic biofuel technology. Going forward, my work addressing limitations to product concentration will address an important frontier in the field of metabolic engineering. Thank you. Now I'll take questions.